Okay, so let's get started. Hello everyone. Welcome to our today's Immunology seminar. It's really my pleasure to welcome our today's speaker, Dr. Xing Wei from Cincinnati Children's Hospital. Um, so uh, I'm actually really a big fan of Xing's work. It's really exciting for me to host him today. Xing is currently a full professor in Division of Infectious Disease in Cincinnati Children's Hospital. So he complete his MD and PhD training in Albert Einstein College of Medicine in New York, and then did his internship and residency in pediatrics in UCSF. So after that, he continued to pursue the postdoc training in University of Washington before he started his independent research program. From 2005, he started his research program, and then soon after, he get a assistant professorship in Department of Pediatrics in Washington, University of Washington. And then sh Shortly from 2007, he moved to University of Minnesota, also in Department of Pediatrics and Microbiology. And then from 2012, he got promoted as associate professor, and then moved to Cincinnati Children's Hospital. To 2016, he became a full professor. And then he was appointed as director of Center of Inflammation and Tolerance in Cincinnati Children's Hospital. Probably a lot of us already learned about Xing's work on the maternal immunity. So Xing's lab are mainly interested in studying to fill the knowledge gap relevant to improving pregnancy outcomes or boosting host defense in newborn babies. They primarily tackle these questions using a immunological focus, addressing fundamental questions like why mothers do not reject their babies, why babies do not reject their mothers, and then how babies adapted during their breast transition from a steroid in utero environment to the external world filled with commensal microbiome at the time of birth. The main projects in Xing's lab are broadly focused on overlapping scientific platforms, investigating how immune responses are regulated with particular focus on the maternal responses to babies, maternal responses to microbiomes, and baby response to mothers, and baby response to microbiomes. Give the importance of the reproductive fitness and pregnancy outcomes in the trait selection. The platform in Xing's lab can also review fundamental new insight for immunology in other contexts, including autoimmunity, transplantation, and cancer. With all the really outbreaking discovery in his lab, Xing was received numerous honors and awards. I'm not going to list all of them, but just a few, including he got the Mead Johnson Awards from Society of Pediatric Research, NIH Director Pioneer Award, Howard Hughes Medical Institution of for Faculty Scholar, and then also Faculty Research Award from Cincinnati Children's Hospital. I personally, as I said, is really a big fan of Xing's work, and his research approach is absolutely spectacular and elegant regarding how these maternal immune tolerance work. It's really great Xing joining us today, and we are very much looking forward to your talk. Please go ahead.
Thank you so much, Dr. Wu, for that introduction and giving me the opportunity to participate today. As you can see, the title of my presentation is called Immunology Learned from Mothers and Babies. And right away, I'll just start off by saying I recognize that this is a overambitious title. Not because we've learned so much of how the immune system works by studying reproduction and pregnancy, but really to highlight the fact that I think we can learn so much more about how the immune system works from this really fundamental developmental windows of pregnancy and early life development. And as was mentioned, my background is as a pediatrician. And so here I want to highlight some of the most recent statistics from the World Health Organization. And what they're talking about here is infant and childhood mortality, or deaths in babies in the first 5 years or 60 months of life. And what they highlight in the yellow parts of this pie chart um, are deaths in the first 1 month of life. And what you can appreciate is that even though that first 1 month accounts for less than 2% of the first 60 months, nearly half of the deaths occur in that first 1 month. And I think what that reflects is the fact that this is really the window of vulnerability for us, okay? That first month after we're born. And so even though I think 5 million is a huge problem, the way that this pie chart lays out, I think it also creates, a huge amount of opportunity. And as was introduced, that I know there's a lot of trainees that might be participating today or listening to this. Imagine what impact you would have in your career if you could take out some of the major slices of this pie chart that cause infant and childhood mortality. Here I want to highlight what I think is always the consistent dominant slices of this pie, which is prematurity and other pregnancy complications. Okay, so in other words, baby are susceptible because of problems related to pregnancy and pregnancy complications. This is the way we take care of babies that are born premature or born through other pregnancy complications here in Cincinnati. And I would imagine in many, many different parts of the world that have really good resources. Okay? But I think one of the difficulties is that this type of taking care of babies in an isolate, in a neonatal intensive care unit, with a lot of tubes and lines for resuscitation, um, even though this can help improve survival of babies, what we're still finding is that there's really high morbidity in those surviving babies that are born through these types of pregnancy complications. And on a practical level, this is also costs a lot of money. So it's not really adaptable to low or middle income parts of the world where there's still the majority of the cases of infant and childhood mortality. So even though I was trained as a pediatrician, we spent a lot of time thinking about not how to take care of babies in this type of situation, but how to improve the health of women during pregnancy and how to improve the health of women before they become pregnant, with the idea that if we can improve upon those parameters, then maybe we can improve upon the health of the babies to prevent pregnancy complications. And you may be wondering, well, something like prematurity, how is it that we don't have any cures for that? This is a review article published now several years ago by one of my colleagues, and what he's highlighting here is the heterogeneity of premature birth or being born too early. And he said it's one syndrome, many causes. And you can see I edited the title a little bit to further highlight how heterogeneous it is. And I think here's a paradox, because if you have something that's so important and then there's many different causes of it, I think you would expect that then many people should be studying it from all of these different types of perspectives. Okay. And for whatever reason, though, I think not a lot of people study it. And I think it may reflect this heterogeneity, because a lot of these types of causes, we don't know what is the driver, and then we don't know what are the secondary causes. And so today I'm going to talk about it from one perspective and try to sort of use that as an attack point to sort of address this issue. And one of the most important parameters of pregnancy is the idea that women can be pregnant more than once, okay, or women can have multiple children, okay. And I think we all realize that because we all um, either have brothers or sisters or know of people that have brothers or sisters. But I think what's interesting is when people study pregnancy, especially from a research perspective, they rarely consider the effects of the prior pregnancy on susceptibility to complications in the next pregnancy. And then here's an example of when it is considered. So this is the Swedish birth cohort where there's over 700,000 women, and they're looking at preeclampsia, which is an inflammatory disorder of women during pregnancy. And they find that in women in the first pregnancy, the rate is about 4%. But if they look at those women who didn't develop preeclampsia in the first pregnancy, then the rate drops to about 1%. And if you further play this out, if you look at women who didn't develop it in the first pregnancy or second pregnancy, then the rate falls even further in the third or fourth pregnancies. And so one of the ways that we've been thinking about how to address prematurity and pregnancy complications like preeclampsia is try to figure out what changed in women after a first normal healthy pregnancy. Because if we can only understand those types of changes that were imparted by that first healthy pregnancy, then maybe we can design therapies to mimic those changes and make preeclampsia pregnancy complications reduced from 4% to 1% all across the board. Here's another similar type of data, but instead of the Swedish birth cohort, this is the California birth cohort. Okay. And here they reported very similar findings, how the rate of preeclampsia in the first pregnancy was about 3%, and then it falls significantly in women with the second pregnancy. But I often like to cite this data in particular is that they did one step further in their analysis because they also looked at what happens in the pregnancies when there's a change paternity or when the second pregnancy was from a partner that was different in the first pregnancy. And what they found is that then the rate of preeclampsia now rebounds back up to the level of the first pregnancy. And I think this is a really important clue because if you think of all the changes that occur in women during pregnancy, there's thousands, tens of thousands of changes on the molecular and cellular level. Okay. But I think with this type of data, I think we may be able to narrow down those changes really to immune cells and particularly to adaptive immune cells, because adaptive immune cells then would be really the only cell type that are capable of distinguishing between fetuses from one partner versus a different partner. Okay. So in other words, it may not have much to do with the uterus expanding and contracting or progesterone, estrogen changes during pregnancy, because none of those should be expected to have the fetal antigen specific features of this partner specificity in these differences. And I've sort of used this as evidence for why we should study um, maternal immune cells during pregnancy and after pregnancy now for nearly 20 years. Okay. And oftentimes I get some feedback that says, you know what, you might be misinterpreting this type of human epidemiology because just because this protection wears off in the second pregnancy with a new partner, there's also many, many other changes that occur when there's a new partner. And for example, one of the most obvious changes is a prolongation of the interpregnancy interval. So the time from the first pregnancy to the second pregnancy. And that makes sense because if if there's a new relationship that needs to start, then it makes sense that there could be a prolongation of that timing. And I think that's where preclinical pregnancy models are really important. And so we've tried to then deal with some of these in our preclinical animal pregnancy models. So for example, if you take mice during primary pregnancy and infect them with a prenatal pathogen like listeria, pregnant mice, just like pregnant humans, are highly susceptible to prenatal listeriosis. This is the rate of congenital fetal invasion, but similar results could be found with fetal wastage or stillbirth. And what's interesting is if you take these mice and let them have a first normal, healthy pregnancy, what you find is that in the second pregnancy, their susceptibility to all of these parameters falls sharply. Okay. So mimicking sort of that protective effects of a first pregnancy against complications in the next pregnancy. And because we're doing this in mice, we can also change the partner from one pregnancy to the other. And so in this case, now if we use a totally new partner, sometimes we call it a third party male, because in the first pregnancies here, we're using males and females that are totally genetically discordant. So now we're using a third party male that's different than the male in the first pregnancy and the female. Then you can see susceptibility now rebounds back up to the level of a first pregnancy. And we've done this in all sorts of different permutations using males and females with totally different haplotypes and prolonging that inter-pregnancy interval, okay? Because in mice, we can control that. And what we find is that it's everything to do with partner specificity, and it has almost nothing to do with the timing in between a first and second pregnancy. And because of this, then we've thought if we can only figure out what makes mice protected after a first pregnancy, then maybe we can apply it to figure out what makes women protected against a first pregnancy and try to design therapies to then mimic those changes. Okay. One of the most consistent T cell types that change in women during pregnancy is the FOXP3 expressing subset of maternal CD4 cells, or called regulatory T cells. This is some of the early data where they plotted the percent FOXP3 cells in the peripheral blood of women before pregnancy and then as the progression of pregnancy. And first of all, you can see there's super wide heterogeneity here. But despite that, the authors of this paper drew an arc that suggests that there's expansion and sustained expansion until 40 weeks term. Okay. And other people have reproduced this data where they were looking at non-pregnant, peripheral blood of non-pregnant women during pregnancy, or women with various pregnancy complications, in this case, preeclampsia again. And what they found is that this normal expansion of maternal regulatory T cells seems to not occur or be blunted in women with preeclampsia. And this is not true just for preeclampsia. Now, many people have described it for other types of pregnancy complications like spontaneous abortion, stillbirth, and prematurity. And now, so many years ago, our lab tried to investigate this, but through the lens of fetal specificity in how the mothers may recognize the baby, because we're after that partner specific changes. And so we devised tools to compare not just the maternal regulatory T cells, but regulatory T cells based on defined specificity. And we asked that when the maternal regulatory T cells expand during pregnancy, is it that cells of every single specificity expand, or is it that cells of some specificities expand over others? And particularly, if those cells that expand would be a fetal specificity. And that's exactly what we found, because when we track the maternal CD4s with fetal specificity, those regulatory T cells expand by 100-fold, 1,000-fold, sometimes 10,000-fold, and really at the expense of all the other specificities. So it seems like when you look at them in bulk, it seems like you see a 50 to 100% increase in the cells, but it's really that it's dominated by the cells that are directly recognizing the fetus. What's also interesting about this type of data is that when we track these cells with fetal specificity in the mice after they gave birth, we find that these cells stick around at much higher expanded levels compared with cells of the same specificity in virgin mice. Okay, and then in a second pregnancy, when they see the fetus again, now they re-expand with much more accelerated tempo and kinetics. Okay, so sort of like how a vaccine works, but instead of targeting effector cells, pregnancy seems to be targeting immune suppressive regulatory T cells that form memory and then are able to re-expand with fetal antigen in a second pregnancy with much more accelerated tempo and kinetics. And so we spent a lot of time figuring out how this works because we think that this may be a way to then reinforce tolerance or protect against pregnancy complications that are mediated by disruptions in the normal tolerance that occurs. Okay. And one of the questions that we began to ask is when mothers immunologically remember their babies through the regulatory T cells, is it true bona fide memory, or could they need some form of antigenic reminders that others have shown for effector CD4 cells? And here's a classic example of that. This is work from one of my prior mentors, Mark Jenkins at the University of Minnesota. And he was tracking CD4 cells that have specificity to Salmonella, foodborne pathogen. And what he found is that after a single oral inoculation of mice with Salmonella, you could find Salmonella-specific CD4s in the spleen or mesenteric lymph node almost for the life of the animal. They stay at expanded levels. Okay. But this persistence of Salmonella-specific CD4 cells also required tonic stimulation by Salmonella, and in this case, in the mesenteric lymph node, because at any time during this course, if you treated the mice with antibiotics to get rid of the Salmonella, then the persistence of these memory pool of Salmonella-specific CD4s would contract. Okay, so implying that memory effector CD4s need some form of tonic antigen stimulation. And so we try to apply this for our findings for maternal regulatory T cells, and we wondered what could be the source of fetal antigen that could be persisting in mothers to allow for memory Tregs to be maintained. And one of the really fascinating sort of considerations is this idea of microchimerism, or the fact that during pregnancy, there's a bidirectional transfer of mother cells into babies and also baby cells into mothers. Okay, and that may not be that surprising to you if you consider that the placenta is not a PVC pipe, but a porous tissue that allows nutrients to pass and waste products to pass. But I think what is surprising is that there's numerous examples now where after pregnancy, these genetically foreign cells seem to persist. So in the example of mothers, people have identified Y chromosome expressing cells on the level of 1 to 100,000 to 1 in a million in the tissue and blood of women who have previously given birth to sons. Okay, presumably then those are fetal microchimeric cells. And the same is true for offspring. Okay, so there's plenty of examples where men or boys contain 1 in 100,000 to 1 in a million cells that have two X chromosomes. So presumably those are maternal microchimeric cells. And so when we want to study how mothers remember their babies, I think the the reverse example is then to study also how children remember their mothers, because all mothers were at one time daughters themselves. Okay, so this type of relationship is a very, you know, simple and elegant one. So if we want to study things in one direction, we can study things in the reverse direction also. And we chose to study this like this because there were already some really remarkable examples that seem to suggest children immunologically remember their mothers. This is Ray Owen. And in addition to many of the work that he did with immune tolerance, he also made this really remarkable example that for women with Rh disease, So women who are negative for the Rh blood group antigen, they're susceptible to sensitization in pregnancy from Rh positive fathers. And when that happens, they develop anti-Rh antibodies so that in the next pregnancy with an Rh positive fetus, then they're susceptible to causing in utero hemolysis by the antibodies targeting the fetal red blood cells. And Ray made this observation that there were some Rh negative women that, despite repeated pregnancies with Rh positive men, they never became sensitized. And he further went on to make the observation that those women in particular tended to be born to Rh positive mothers themselves. Because they were Rh positive, then Rh became in those women what we would call a non-inherited maternal antigen. And then they were tolerant to it so that despite repeated pregnancy-induced sensitizations, they were never sensitized, so they were tolerant. Another example of that type of tolerance is work from John Van Rood and Franz Klaas. And what they observed in studying people with kidney failure is that, so this is before the days of recombinant erythropoietin. So people with kidney failure required chronic transfusions. And what they observed is that through these people that were repeatedly getting transfusions from multiple different types of donors, they never became sensitized to the non-inherited maternal alleles. Okay, as another example of children maintaining tolerance to their mothers. And then finally, I'll just highlight this is work from Will Burlingham at University of Wisconsin. He was also looking at people with kidney failure, but in a totally different context, in the context of sibling to sibling transplantation. And what he observed is that when you have matching between siblings, donor recipients, and kidney transplantation that's matched for the non-inherited maternal HLA haplotypes, then the long-term survival of those kidney allografts is almost identical to an HLA identical sibling to sibling donor, whereas there was no protection when you're matched for the non-inherited paternal allele. So several examples here that children are actually much more tolerant to their mothers compared with their fathers. A few years ago now, we proposed that one of the reasons why this type of tolerance may be conserved is not to have allografts like kidneys stay longer, but maybe it would have a reproductive advantage, and especially a cross-generational reproductive advantage, because in a Mendelian inheritance, women can only share one or the other of their alleles. But wouldn't it be interesting if through Mendelian inheritance, they shared one of the alleles, in this case, either green or red, but through transmission of the microchimeric cells or this tolerance phenotype, they can also share the other allele. So in this case, by transmitting the red allele through traditional genetics, they could also transmit tolerance or a reproductive advantage during the next pregnancy if they encountered a green male that shares the non-inherited maternal antigens. And that's exactly what we showed, because in our animal models of fracturing fetal tolerance, it was nearly impossible to do so when we generated breedings where we had this matching between non-inherited maternal alleles and the offspring. You may think, though, that in nature, this may not happen very frequently. So besides for this reproductive advantage, could there be some other advantages for people maintaining microchimerism or their mother cells? And one of the things we thought of is whether or not presence of phenotypically different microchimeric cells, how they could impact severity of autosomal recessive disorders like cystic fibrosis, hemoglobin SS disease. And in this case, we tested it with a disorder called complement deficiency. So as you may know, complement is a blood protein that protects our bodies against infection. Um, People with complement deficiency are highly susceptible to infection, and especially infection by bacterial pathogens, and so are mice. And so when we take complement deficient mice, we can generate them in two different ways. We can breed them complement deficient by complement deficient parents and generate complement deficient mice, or we can generate them by those born to complement heterozygous mothers that are mated with complement deficient males. And in that case, we have maternal microchimeric cells that are complement producing. And what we find is that the repertoire of microchimeric cells is sufficient to increase levels of complement in complement deficient mice substantially. And in this paper we published last year to show that this offsets their susceptibility to infection. And we know that this level of complement is produced by the microchimeric cells and not from breastfeeding or in utero transfer of complement, because we're able to develop tools where then if we deplete the microchimeric cells, then we lose the complement, and then we override the protection against infection. So many reasons for people to be immunologically very complicated and contain this repertoire of microchimeric cells, not just from a reproductive benefit, from several other advantages. Because of this, we propose an interesting new word in the literature. We called it the microchiome, okay, analogous to how people have genetically foreign microbes living in our bodies in mucosal barrier tissues that we recognize are immunologically advantageous. We wondered if these microchimeric cells may be doing the same thing. So we called it the microchiome of cells. And you can imagine how complicated it would be if you imagine this lady. She would contain the cells from her mother inside of her. In a first pregnancy, she would be seeded by fetal microchimeric cells. In a second pregnancy, by a new wave of fetal microchimeric cells, and in subsequent pregnancies. And then we began wondering, how would these different types of microchimeric cells interact with each other? And would they synergize with each other? Would they fight with each other? And maybe this could explain the immunological complexity of women compared with men. So we designed some experiments to test that, and we found something quite interesting. So when we took mice that had defined types of maternal microchimeric cells, and when we got them pregnant again, what was super interesting is that they seemed to lose all the pre-existing pool of prior maternal microchimeric cells, and they were replaced completely by fetal microchimeric cells in the next pregnancy. And with that replacement, they also lost the memory of their mothers. They lost the tolerance to their mothers by all the parameters that we can test for. So we wrote in this paper that daughters seem to forget. Okay. And what was interesting about this relationship, though, is when we tested how mothers would remember their babies. So in this case, an orange lady remembering blue babies from a first pregnancy. What was interesting in the second pregnancy, if she encountered a brown baby, she would still completely lose all the fetal microchimeric cells from that first pregnancy. But immunologically, she never forgot that baby, meaning that she still retained tolerance to those fetal antigens, despite intervening pregnancies with different partners. And in this paper, we iteratively put female mice through multiple successive pregnancies, often with different males, and we found no matter how many successive pregnancies, they never seem to forget that mothers accumulated an advantage to always remember all of their babies. And I think this teleologically may make sense, because while each person only has one biological mother, women can remember women, because they can have multiple pregnancies and multiple children, there may be an advantage for them to remember all of their children. And immunologically, we showed that in both cases, when you lost either fetal microchimerism or maternal microchimerism, you led to contraction of that antigen-specific pool of regulatory T cells. But how this was distinguished is that when mothers lose fetal specific Tregs through displacement of their cells, those CD4s undergo a very special differentiation program, what we call X-Tregs, because when they see that antigen again in the context of another pregnancy, then they quickly re-express FOXP3 with much more accelerated tempo and kinetics. That's very different than how T cells behave when they lose stimulation by maternal microchimeric cells. Okay, so just in the first part of this presentation, I would say for how immunology learned from mothers and babies, I would say one important point is that one of the foundations for how we think about immune tolerance is this concept of binary self versus non-self antigen distinction. And by studying it, though, how it works in this context of reproduction and pregnancy, I think we've sort of extended the antigens that we're tolerant to beyond just self antigens, but also hereditary antigens that are expressed by these microchimeric cells, or asking the question of considering tolerance if individuals are not just binary self and non-self, but are constitutively chimeric. I'm going to just take a sip of tea, and then we'll pivot to the next section, okay? Thank you so much. Okay, so I introduced this type of research with these statistics from the World Health Organization, and I sort of told you guys the importance of pregnancy and pregnancy complications in pediatrics and in causing mortality in the most vulnerable population. In the last few minutes of this talk, I want to pivot a little bit and sort of highlight the fact that many of the other slices of this pie is really infection, okay? And this is the second killer outside of pregnancy and pregnancy complications for newborn babies. And then some of the work that we've been trying to address this. And I think one of the key questions is, well, then, why are babies more susceptible to infection? Okay. Turns out that just like human babies are more susceptible to infections, so are baby mice. Okay. This is infection susceptibility for 5-day-old neonatal mice to Listeria monocytogenes, a common neonatal pathogen as well as perinatal pathogen. And you can see if you infect these neonatal mice, the majority of them die from infection, and adult mice don't. And when neonatal mice die, they're chock full of the pathogen that you infected them with. And it has nothing to do with the fact that baby mice are smaller, because if you mass adjust the inoculum for adult mice, then adult mice are still completely resistant to infection, unlike baby mice. Okay. One of the explanations that I think we still are teaching medical students is that, well, maybe this susceptibility of newborn babies reflects immunological immaturity or immaturity of their immune cells. Just like their lungs may be immature, their intestines may be immature, their brains may be immature. Maybe their immune cells are immature also and require some period of postnatal maturation to acquire adult-like function. And some of the data to support that is that if you take cord blood cells, so cells from newborn babies, compared with adult cells and stimulate them, they're always hyporesponsive. Okay, here's an example where we're stimulating them with LPS, and you can see that cord blood cells are hyporesponsive in producing pro-inflammatory cytokines that are protective against infection, like interleukin-6, TNF, or interferon, compared with the same adult cells. And this type of hyporesponsiveness has been linked with functional immaturity of neonatal cells. And so many years ago, our lab said, if this is the case, we want to figure out the most important immune cell in babies that's immature, because if we can boost the maturation of those cells, then maybe we can boost immunity against infection. And so we designed these very simple-minded experiments where we took neonatal mice and then we just shoved unfractionated immune cells from adult mice into them. Okay, just transferred them intraperitoneally. And then we said, if we loaded them up with a bunch of adult immune cells that are mature, then could we rescue that infection susceptibility in babies? Because if we could, then we can start fractionating the subsets of adult immune cells that we transferred to figure out what are the most important cell types that are immature. And to our probably frustration at that time, we couldn't do it. No matter how many adult immune cells we parked into baby mice, they still remained highly susceptible to infection. Very different than adult mice, and then almost similar to neonatal mice that didn't contain any adult immune cells. And we were quite puzzled and frustrated with this result until we started looking at the activation of these cells. Okay. And what we found is that adult immune cells in response to infection became activated. They produced TNF alpha and interferon gamma, similar to peripheral blood and cord blood cells. Neonatal cells were hyporesponsive, just like cord blood cells are. But what was really interesting is these adult cells that we parked into the neonatal mice that we can distinguish based on differences in chondritic marker also became hyporesponsive, just like the neonatal cells. And this made us challenge the idea that infection susceptibility in babies was really hyporesponsiveness or immaturity, but instead we proposed this idea that maybe newborn babies' immune systems are actively suppressed. And that's what causing the hyporesponsiveness or infection susceptibility. And I think one of the most important reasons why newborn babies' immune systems has to be immune suppressed is to allow that transition from a near complete sterile in utero environment in utero to the external world filled with commensal microbes at the time of birth. And here I want to remind you that when babies are born, it's not like in a beautiful hospital wing where there's clean sheets and clean bedding waiting. Okay, most mammals are designed to be born in the manger, okay, where there's an extremely dirty environment, where they would transition right away to a world that's very dirty and filled with commensal microbes right away. And we showed some data that if you lack that immune suppression layer in newborn babies, then you would have catastrophic commensal microbiota-driven inflammation at mucosal barrier tissues. And this was published, so I invite you guys to look at that. But I will say that this type of data then did challenge the field, because many of us had been trying to design vaccines that we could give to newborn babies, okay, very soon or shortly after birth, with the idea that if we can boost immunity in that very early developmental window, then maybe we can reduce this window of susceptibility. But we also, when we realized that babies were actually operating under a layer of immune suppression that's doing a greater good, then it made us challenge our ability to do this. And so instead, many of us have now focused on, instead of vaccinating babies as soon as they're born, to vaccinating mothers or reproductive age women, with the idea that if we can prime immunity in them, then maybe we can have that immunity be naturally transferred from mother to baby that then maybe would be a more efficient way to offset this window of susceptibility. And this is some cartoons that we wrote in a review a few years ago. And most of the vertically transferred immune components, or the best characterized, are in the form of vertically transferred antibodies. And I think this creates a natural conundrum because, as you know, antibodies um, do very effective protection against bugs that live outside of the cell or in mucosal tissues, but they're largely ineffective against bugs that live inside the cell. And that's because antibodies can never cross the plasma membrane of cells. And that's why instead of antibodies, we also need T cells, which are then much more effective at killing intracellular pathogens, with the idea that antibodies and T cells do complementary but non-overlapping things with regards to host defense against infection. But I think then this creates this conundrum because then if only vertically transferred immunity is transferred through antibodies, then this would create naturally occurring host defense in babies. My daughter always gets embarrassed when I show this. This is when she was born. She's 12 years old now, and she didn't say this, but I wonder if this is what she was thinking about. That if she were only getting antibodies, then maybe she would need protection against intracellular pathogens as well. Um, we we particularly wondered if this could be transmitted through these microchimeric cells, and if microchimeric cells could also exist in the form of cytolytic CD8 T cells that would protect against infection. And to study that, we used Listeria monocytogenes, not only because it's a neonatal pathogen in humans and mice, but more importantly, because the intracellular life cycle of this bug had been worked out so well, where protective immunity was known to be exclusively mediated by T cells with little or no role for antibodies. So we designed what I thought were simple-minded experiments where we took mice and gave them a sublethal Listeria infection, let the infection resolve. And like many of you who might have done this or read papers of people who do it, where then you track the Listeria specific T cells as a way of reading out the T cell response to infection. We didn't do that. We took these mice and instead mated them and then challenged their babies with the idea of then we wondered to see if there was vertically transferred protection against a classical intracellular pathogen. And sure enough, there was. So babies born to mothers that had prior preconceptual Listeria infection were always much more protected with lower CFUs and improved survival. But what was interesting about this data was that we could not consistently find Listeria-specific T cells in these offspring, um, um, but we could always consistently find Listeria-specific antibodies in the offspring. And one of the most definitive experiments that we did is that when we used mothers that were B cell deficient or couldn't produce antibodies in this context, now this protection that's vertically transferred was completely lost. And this wasn't because the pups needed B cells, because we sired these pregnancies by wild type males. So the genotype of the pups was heterozygous or functionally wild type. But you can see the loss of anti-Listeria antibodies that's vertically transferred led to complete loss of protection in this context. So it seems interesting that somehow in the context of vertically transferred immunity, Listeria was breaking all the rules, that now antibodies seem to be able to protect against an intracellular pathogen. We moved this, instead of just having it be vertically transferred, we call this horizontally transferred. So we took serum from Listeria prime pregnant mice or virgin mice, and we showed that you can adoptively transfer the serum from pregnant mice, but not the serum from virgin mice to be protective. And what was really interesting is if you took this non-protective virgin serum and then transferred it, instead of directly to pups, if you transferred it to a pregnant mouse and then have the pregnant mouse transfer it to the pups through vertically transfer, now this virgin sera could acquire protective features. And so we were really interested to figure out what changed in pregnancy to allow what seems like non-protective antibodies to acquire protective features against intracellular pathogens. Now, just quickly say that we generated a lot of negative data. Okay, the titer of the antibodies was exactly the same. Their isotypes were the same. And there was always their glycosylation was the same also. But when we sent these antibodies to a collaborator, they always gave us the feedback that the virgin antibodies or non-protective antibodies seem to always run a little bit heavier on a mass level than the pregnant antibodies. And it was about 40 Daltons. Okay. We didn't totally understand what that meant until later when we realized that those virgin antibodies always contain on their terminal sialic acid, so this blue diamond, one acetyl group, so 6 molecules or 42 Daltons. Okay. And this acetyl group turns out to become deacetylated during pregnancy. And this 6 molecules turns out to be life or death for the mouse because when it becomes deacetylated, then it becomes a ligand for CD22, which is a negative regulator on B cells. And as you may know, the neonatal environment is loaded with these IL-10 producing B regulatory cells. And then when you suppress them from producing IL-10, that's one way where you can unleash protective immunity through vertically transferred antibodies. Okay, so this is a table in Janeway that we showed earlier. You don't have to do it to your textbook, but I would say in some ways by studying how immunology works in this concept of through vertically transferred immunity, then we're able to amend it by also showing that protection against intracellular pathogens can also be transferred by easily be mediated by IgG, but IgG that's modified during pregnancy. Okay. So lastly, I just want to conclude by saying, what I started out by saying, that there's a lot of immunology to be learned from studying pregnancy, mothers, and babies. And I think we're just at the tip of the iceberg. And I was delighted to share with you some of the key observations that we've made now over the past, 10, 20 years using these types of concepts. And then these are the people in my group that did the work that I shared today. And obviously, I'm super grateful for our funding agencies and for all of you for giving me this opportunity to talk about this. And happy now to take any questions or discuss anything.